Hello, and how are you? My name is Winnie Barawa, and welcome to this session on ribonucleic acid, commonly known as RNA. If you've been following, I've been doing lectures on anatomy and physiology, and if you want to follow more, check on our playlist on anatomy and physiology on this channel. So in this session, we're going to analyze the RNA, its characteristics, its structure, and how it is formulated. That is the synthesis. So stay with me all the way to the end, and don't forget to subscribe if you're new. This is a channel that we do lectures, lessons, and expert interviews in relation to healthcare. If this is something that is of interest to you, please click on the subscribe button and notification button so that you'll be let known when we give the next lectures. So RNA, where is it found and how does it come about? So to recap, just the way we did under the DNA, this is how it happens. In the cell, within the cytoplasm, we have the nucleus here with us. And it is in this nucleus that we mainly have our DNA. So DNA is double-stranded, it is helixed, or it is coiled in a way. But for us to get an RNA, the RNA will be generated from the DNA through a process known as transcription. So because the RNA is single-stranded, what happens is the DNA will temporarily open up, create for us the RNA, then the RNA template will move from the nucleus and get out via what we call nuclear pore and be taken to the ribosomes for protein synthesis. Now, the process of translating our RNA to form amino acid is what we call translations. Okay, so this is how the RNA comes about. It's a complementary copy of a DNA strand. So here is a summary of this where we say these two, that is DNA and RNA, are important for coding and decoding genetic information. But the main bit that does the decoding is the RNA. So the RNA can code and the RNA can also decode the genetic information. So who creates the other? We've already said the DNA codes for the RNA. Then this RNA strand will be used to code for our amino acids that will be used as building blocks for formation of the protein. So moving forward, this is what we mean. So we have our nucleus, which is covered by the nuclear membrane. And this nuclear membrane has pores, what you're calling the nuclear pores. So in the nucleus, we shall create the RNA template. We will come later on to see the different types of RNA, and as what you're seeing, it's written as mRNA, what we famously known as messenger RNA. But that's beyond the point for now. For now, we are focusing on where does the RNA come from. So the RNA, in simple terms, we can say it's a complementary copy of our DNA. The only difference that varies with the DNA is that our RNA has uracil and our DNA has thymine. So that's how these two templates will vary. But the main bit is the RNA is born from the DNA, where our DNA opens temporarily, releases the RNA, and the RNA can travel out of the nucleus and gets out to the nucleolus and head to the ribosome, where it can help us with protein synthesis. So the formation of RNA is through a process known as transcription. However, while the RNA moves to the ribosome, we will need to translate that template to generate amino acids. So the decoding of this template is what we call translation. And then the forming of the amino acids from this RNA is generally what will formulate the protein synthesis here. So see how interesting it is? All right. So let's look at the RNA structure. So the RNA has basic chemical compounds, what we also call the nucleotide. And this is the formation or the backbone that will be used in bringing out our RNA structure. The basic structure here will have the phosphate or phosphoric acid. We have a sugar known as the pentose sugar that we call the ribose, and then we have 4 nitrogenous bases, which contains adenine, uracil, guanine, and cytosine. So here's the basic structure of our RNA. We have a phosphate group, so what you call the phosphate, phosphoric acid. We have a pentose sugar, what you call the ribose, and then we have a base. And these bases can be different types of bases. So here we can have the 4 of them. 8 of these can be used for the structure. So there's an adenine written as an A, uracil written as a U, a guanine G, cytosine the C. So those are the 4 main bases here. All right, let's move forward. So how does this RNA come about? What you are now saying is the RNA synthesis. So RNA is generating through a process known as transcription. And the transcription, it's a process that occurs in our DNA in such a way that the DNA will open up temporarily to form the RNA, and this RNA will come out of the nucleus and go into the cytoplasm, heading to the ribosomes for the purpose of forming a new, or for the purpose of forming a template that will be used for formation of amino acids. So how does it work? So here we have our DNA strand or double helix, that normally opens up or unwinds. Then you need to remember that this unwinding is very temporal, or it's temporary process. So it will unwind to give us 2 template strands, whereby one template is the coding template, and the other template, what you are calling non-template, will be the non-coding template. So the template that will be coding, for example, here we are using a single template, it will help us to generate the RNA copy. So this will be a complementary copy. That's what we call a complementary copy. So it will move, we normally say the DNA codes from the direction of 3:5. So that's why you find, from the 5:3 sequence. So we have this sequence here where now we will start coding it. So the RNA will follow the same sequence. And because we are using the coding strand or the template strand, we have what we call the promoters. Promoters, these are enzymes or codes that will help us initiate these processes. So we will code, but keep in mind that where there is an adenine, we will pair it with a uracil, because in RNA we do not have thymine, we have uracils, and you will get this code. Then this code will get out, or this RNA template will get out and go to the cytoplasm, where it will be used now for the coding of amino acids. So by description, to summarize this, is that the RNA is a single-stranded molecule, and for us to synthesize the RNA, the 2 strands of the DNA molecule, they will unwind, or they will separate temporarily. And one of these DNA strands will then be used for the synthesis of the RNA strand or the RNA molecules. So here is what has happened. These 2 strands have opened up temporarily, and we are creating a template of the RNA. So the RNA will create codes, that is the code triplets of the DNA, creating what we call complementary code triplets, the codons in our RNA. And these codons in turn will then be used to help us control the sequence of amino acids in the protein synthesis process that happens now in the cell membrane. So while this transcription is happening, there's a manner of pairing that is supposed to happen, or there's a manner of base pairing that is supposed to happen while we are generating this template. So what happened is, we are using the DNA template to generate a complementary template, that is the RNA template. So what happened is, where in the DNA we have guanine, on the RNA we shall have cytosine. Where in the DNA we have cytosine, in the RNA we shall have guanine. Where we have adenine on the DNA, we shall generate a uracil on the RNA. Why? Because we cannot pair adenine with thymine in the RNA. The RNA does not have thymine. Instead, it has uracil. So where we have thymine in the DNA, we shall just normally have the adenine because the RNA has adenine. So the thymine is normally replaced with uracil in the RNA bases. The same way we had hydrogen bonds in our base pairing is the same way we shall have also hydrogen bond while we are creating this RNA template. Why is it so? Is because the hydrogen bond is very weak and it can easily break to release our RNA from this DNA template and travel off from the nucleus to head to the cytoplasm where it can participate in the protein synthesis. And so this is how the template comes about. So we have our normal DNA here, double helixed, well structured, organized in the proper sequences, that is 5:3, 3:5. When this DNA opens up temporarily to generate for us the RNA, we will have the coding template of the DNA and the non-coding template of the DNA. When this is done, we shall therefore have the RNA template that can be transported out of the nucleus and head to the cytoplasm. So this is the RNA template or the RNA complementary template that moves out and heads to the cytoplasm for coding. So in this case, we are saying in the RNA, we do not have thymine, so you will not see any C in this template. Instead, you will see, instead of the T, you will see uracils. So before I said there are different types of RNA, and the first one that we have seen in our description is the mRNA, famously known as the messenger RNA. So the RNAs are mainly three, where we have messenger RNA, transfer RNAs, and the ribosomal RNAs. So let's start with the messenger RNA, which is famously known as the mRNA. The messenger RNA, what it does is it's the main one that gets produced from the DNA. Generally, when this transcription process has happened successfully, what we produce there is mainly the messengers. But the messenger RNA has the main role of carrying the genetic code from the DNA or from the nucleus, to the cytoplasm for the purpose of controlling the type of protein that are being formed. So it is the main one that gets to be coded. And then the second one is the transfer RNA, the tRNA. What this tRNA does, it mainly participates in transportation. So it transports our activated amino acids, take them to the ribosome, and allow them to be used for protein synthesis. So that's what the transfer does. It's a transport mechanism. It's a transport channel. So it takes our amino acids, or it takes our activated amino acids to the ribosome where they can be used for protein synthesis. And the last one is the ribosomal RNA. Ribosomal RNA, it works together with other proteins in the body to create our ribosomes. So this generally participate in being or in forming part and parcel of the ribosome. They're like a part of the structure of a ribosome. So they're mainly RNA used in the purpose of forming the ribosomes. So to avoid all these stories, let's see how they look like in a diagram. So this is our summary of the RNAs. So I've said we have the rRNA, the ribosomal RNA, the transfer RNA, and the messenger RNA. So let's start with the rRNA, where we say they're called the ribosomal RNA. The ribosomal RNA, basically we say they form part and parcel of the ribosomes, or they're part of the structure of the ribosomes, together with other proteins, while the transfer RNA is responsible for taking amino acids to the ribosome to help us with forming the proteins. While the messenger RNA, it's part of the copy of our DNA, and it's mainly responsible for the purpose of being transported for translation and to help us create the proteins. So how do they look like? The messenger RNA most of the time looks like a strand, so it's used to encode proteins. The transfer RNA is an adapter or a transfer channel of taking our amino acids to the ribosomes for translation. And the last bit is the ribosomal RNA, which is part and parcel of forming our ribosomes. So to finish up, what is the difference between DNA and RNA? So there are similarities and then we have the differences. With similarities is that both of these nucleic acids, they are known as nucleic acids, of course. They are composed of nucleotides. They have the pentose sugars as their backbone, and they have 4 different types of bases. So here are the similarities. They are nucleic acids. They are made of nucleotides. They have phosphates or the sugar phosphate backbone, and they're made up of 4 different types of bases. While on the differences between DNA and RNA is the DNA is found in the nucleus, while the RNA is also found in the cytoplasm, although you'll also find it in the nucleus. DNA is part of the genetic material, while the RNA is an assistant to the DNA. It does not mainly form the main part of the genetic part, but it assists the DNA to carry forward these genetic characteristics. The DNA has deoxyribose, the RNA has ribose sugars. The DNA has the bases of adenine, thymine, cytosine, and the guanine, while the bases in RNA has the same as the DNA bases, but they include the uracil. The DNA is double-stranded, while the RNA is single-stranded. The DNA is used purposefully for transcribing to give the different variety of RNA molecule, while the RNA is more important in participating in translation that will be used to make proteins. So I hope you're able to distinguish these two now in details. So thank you so much for staying with us all the way to this side. And if you like this content, you can give us a comment, share these videos with your classmates. Remember to subscribe also and hit on the like button if you like our content. Thank you so much and see you in our next class. Bye bye.